This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 543, recorded on April 12th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Good afternoon, Vincent.
Who has a little rough throat.
Yeah.
As those of you who listen to TWiP this week will already know.
The kind that make you sick.
The kind. Where did you get that?
No, I think I got it off the handrail of one of the down or up
I told you
stairs.
not to lick the handrails. Why did you do that?
Now, you told me too late.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here in Ann Arbor, it is, I just lost it. Let me get it on a different channel. 59 degrees Fahrenheit.
Oh, that's
and
lovely.
so that's pretty close to probably about 15 Celsius. So.
But you also have a cold, you told me.
Yeah, I do.
And you didn't get that from the subway handrail.
No.
I don't have subways in Michigan, in Ann Arbor, right?
No, nor in Georgia, where I think I might have gotten it.
Well, they have the MARTA, right? That's kind of a commuter rail, right?
Yeah, and actually in the airport, there were 3 different train possibilities, and I took 2 of them to get to the rental car place. So, yeah.
Yeah, you do have a little congestion. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's a little warmer here in Madison. It's 65 degrees Fahrenheit here. And I actually don't have a cold.
Oh, would you like one?
Not at the end of the semester, no.
No, not a good thing.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 45 Fahrenheit, 7C, overcast, kind of cloudy and cold. I do not have a cold, and I have ridden the subway in Georgia, in Atlanta.
And from Austin, Texas, Rich Condit.
Hi, everybody. 71 degrees and sunny. That's 22 Celsius. It's a glorious day.
Cool.
You went sailing last week, right?
Last week, it was not on. Well, last week, I don't know. I've lost track of time. But yes, I have been sailing since I talked to you guys last. That was glorious.
When you look up the definition of emeritus professor, that's what it says.
Yeah, it says
I've lost track.
I'm not going to ask you, Dickson, to read anything today. Okay?
Please don't. I could do it, but it'll be
not because I don't want to, but you asked me to before. So.
Yeah, I think for the sake of the listener audience, you shouldn't.
Kathy, are you there?
Yes.
I would like you to take that first follow-up, please, from Bradley.
Okay, from Bradley. Dear Vincent and crew, I was driving from Princeton to New Brunswick, New Jersey, of course, when I heard the short background to TWiV 541 and heard Kathy read. The initial description of mitoviruses was accomplished through research on 2 related fungal pathosystems that are often confused by those with only passing familiarity in plant pathology. I thought, geez, that sounds like something I would say. And sure enough, it was, from my AVR review on narnoviruses and mitoviruses a few years ago. I am thrilled that you have taken a continued interest in mitoviruses. I was slightly disappointed that the paper you chose to highlight in TWiV 541 was Max Nybert's largely bioinformatic 2018 paper. Slightly disappointed because Max is terrific and it's a nice paper, but highlighted in the current issue of JVI was this paper from Massimo Torina's lab in Turin that takes the plant mitovirus work a step further and describes experimental results with one of the viruses. And he gives a link. The paper Massimo's group from Massimo's group answers some of the questions you and the others posed in your discussion and would be worth visiting. A quick elevator summary of mitovirus discovery. A group led by Ken Buck, a true pioneer in fungal virology at Imperial College, showed in the 1980s that diseased strains of Dutch elm disease fungus, Ophistioma novo-ulmi, contained double-stranded RNA that was consistent with mitochondrial inheritance, maternally inherited. Then in the early 1990s, a postdoc in my lab and I showed that a similar element in the chestnut blight fungus, Cryphonectria parasitica, exhibited maternal inheritance, mitochondrial localization in fractionation experiments, and an RNA sequence that made sense only if you invoked fungal mitochondrial codon usage, UGA equals tryptophan, among other properties, and he gives a link. From there, the floodgates opened, and more than 100 such elements have since been characterized in fungi, but relatively little progress has been made. As you alluded to in your conversations, I think Alan made the point, mitochondria are hard to work with. We effectively demonstrated that through years of failed experiments, with some good successes. Regarding the name, I think I named both the new family Narnaviridae, naked RNA virus family, NA-RNA, and mitovirus genus, self-explanatory, when we erected those taxa for ICTV for the 6th report when Reed Wickner was fungal virus subcommittee chair immediately before I was. And yes, regarding Narnaviridae, I thought of the Chronicles of Narnia too, but it seemed to beat the competing suggestion Nudiviridae, which I thought sounded even sillier, and was nevertheless taken subsequently for a family of DNA viruses. Fast forward to now, some progress on a couple of fronts. First, a bioinformatics study by Max that came out a bit earlier than the paper you discussed showed that mitovirus UGA, tryptophan, codon usage parallels that of host mitochondria. It must show that. That's the title of the paper. In other words, if you're a fungus, the fewer tryptophan encoding UGA codons your mitochondria have, the fewer your mitoviruses have. Makes sense, right? And second, in a recent JVI paper that you referred to, Vincent, Nobuhiro Suzuki and colleagues, I'm on the paper, but they did the real work, showed that we could move mitoviruses among both closely related and more distantly related fungi by moving their mitochondria via protoplast fusion and repeated rounds of selection. And he gives a link for that. Still difficult, but this should open a few doors. Keep up the great work. I love the podcast. It helps me keep my no NPR 2016 to 2020 pledge, and I am constantly impressed by your collective scope and complementarity. And I especially enjoy it that you guys grope your way through a plant virus paper such as the eLife paper from Stefan Blanc's group in TWiV 539 with admirable enthusiasm but limited understanding of plant anatomy and biology. Reminds me of the ancient Indian parable of the blind man and the elephant. And I hope you keep your interest in mitoviruses and fungal viruses more broadly. There's a lot going on in the field, though sadly limited funding. Best regards, Brad. And he is professor of plant biology and pathology at Rutgers and director of research of the New Jersey Agriculture Experiment Station.
So who would fund this work?
USDA.
Yeah.
Maybe NSF.
Could be maybe NSF.
And you know, depending on the fungi you're working on, NIH.
Yeah.
True.
Yeah. But mostly they work on these.
Mostly they work on these plant fungi.
Yeah.
So it would be USDA, NSF.
And then
USDA has money, but they spend it on other things.
Right. And it depends on the crop also, because there are associations for corn and soybeans and stuff like that.
Well, Brad, thank you, and come by sometime.
Yeah.
Pop in if you're ever here. And
not far.
And we can have him on a plant virus show so he can get the plant biology straight for us.
Yes.
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 Well, you know, the more we do, the more we learn, right?
Mm-hmm.
So
I have to go back to school to get plants right. You know.
no, this is school right here. This is TWiV school.
Okay.
This is school.
Okay.
All right. Admin writes, I think scala means staircase, maybe like a spiral staircase or from low tones to high tones. And the weather in Pensacola is mild but overcast today. I love the weather on TWiV and, of course, the virology. That meant to say in the previous email, how cool this guy, Massimo Turina, is in Turin.
Yeah.
Yes. I noticed
like that too.
Of course, it's Torino in Italian, so it's not really so close, but it's cool anyway. So I could be Racaniello from Racaniellia.
I think you'll have to found the town of Racaniello.
There's no town. Alan, can you take the next one?
Sure. Les writes, this is referring to Weekly Science Picks and my cherry blossoms, my snarky cherry blossoms pick. A biotech pioneer funded the capital's cherry trees. Takamine Jokichi funded a gift of 2,000 cherry trees from the mayor of Tokyo to the city of Washington. I think it was his idea. He was a pioneer of American biotechnology in the 19th century. He discovered adrenaline and a critical diastatic enzyme. Yeast can't digest starch in grains. The enzyme converts starches to sugars. He had the first patent on a microbial enzyme in the United States. Being Japanese, despite living in the United States for over 20 years and marrying a citizen, he could not become a citizen himself.
I think this is familiar. I think we've maybe done this many episodes ago, but very interesting.
Yeah.
Rich writes, guys, isn't that Rich Condit, right?
No.
I know. Once again, many thanks for a very interesting show, which was thought-provoking. It got me thinking as to the difference between a virus and a plasmid. Good question. Apologies in advance if I had missed something obvious, but both replicate using the host cell's machinery. Viruses are normally separately packaged in a capsid, but this does not apply to the mitoviruses in TWiV 541, and Hep D relies on Hep B capsid proteins. Also, neither exit the cell, so the host-killing virus rule does not apply. Is the difference that mitoviruses encode RNA polymerase, so they are masters of their own destiny, and that plasmids do not exist in fungi? I've seen references to fungal plasmids. So if a plasmid were to encode RNA polymerase, it would be classified as a virus. Mitoviruses appear similar to viroids, and some viroids replicate in plant chloroplasts, per Wikipedia, the Avsun viroidae. So are mitoviruses really a type of viroid? A possible Talmudic question, perhaps. Cheers. Oh, so this is apt because we had a textbook meeting yesterday in Princeton, and we were going over the chapter on viroids and satellites. So, the mitoviruses are circular RNA molecules. They don't have a capsid. They are called viruses. They don't even leave the cell. Viroids are circular RNA. They code for nothing, but they're called viroids, and I don't think one open reading frame should make a difference. So I would say viroids are actually viruses. It's just that they were discovered in the 60s before we thought of such things, so they were just called T.O. Diener thought they were something different, so he called them viroids, but I think they're all viruses. I think satellites are viruses. The real question is, so there are two things. One is, do you need a capsid to have a virus? And I think the answer is clearly no, because mitoviruses don't encode capsids or viroids. So what's a plasmid?
Yeah.
A plasmid has been defined as a DNA molecule that replicates outside of the chromosome, right?
Right.
Sounds like a virus to me.
An RNA is not a plasmid by that definition, but I guess, I don't know. It's not. So can we say RNA molecules that are autonomously replicating are not plasmids? I guess not. So circular RNA molecules made by backsplicing in eukaryotic cells, those are not plasmids. They're just circular RNA molecules, right? So
are they viruses?
The question is, so viruses, a property of viruses is that they're obligate intracellular parasites, right? So I don't know, these circular RNAs don't seem to parasitize cells. Anyway, what about, does anyone know of DNA elements that are virus-like, which don't encode capsids, which just replicate autonomously and spread from cell to cell? Oh, those are called plasmids.
Plasmids.
Although in eukaryotic cells, we call them extra chromosomal elements, but, right?
They're plasmids.
They're plasmids. And plasmids do exist in fungi.
Double-stranded DNA plasmids?
Yes. Or at least you can do lots of fungal transformations, which I have learned is called transformation and not transfection, with lots of plasmids.
Well, so the thing is, a plasmid does multiply, and it goes from cell to cell as the cells divide, which is kind of like these mitoviruses do in the narnoviruses, which are the cytoplasmic equivalents. So it's a fine line, right?
Yeah.
Yeah, it's a really gray area. It's almost, I get the feeling that it's almost historical. If plasmids had somehow remained obscure until today, and we all of a sudden discovered them, we might call them viruses.
Yeah.
Yes.
That's right.
But they've been around since before we knew about things that we now call viruses that don't have capsids.
Mm-hmm. Yeah, I think
it's
you're
like
right.
why we call the Missouri River the Mississippi River. It's the order we discovered them in.
Yes.
And we draw these distinctions. This is a virus, this is a plasmid, and then we discover that biology doesn't draw these distinctions. So,
Oh
yes, this is all for us.
Yeah,
and by the way,
for our use.
By the way, in this letter, there he has an emphasis on whether or not a given element encodes a replication enzyme, and there are plenty of examples of DNA viruses that don't encode a frank RNA polymerase or DNA polymerase.
Oh, yes.
Well, they all encode
or a polymerase with any other name.
Right. They all encode something that will direct the attention of the cellular replication machinery to the viral genome. But plasmids do the same thing. So there's no distinction.
I think your point is good. That's a historical thing. Yeah. Because if you read Kunin's papers, he talks about selfish replicating elements, right? But we would call them viruses, really. They go from cell to cell, they replicate. They don't have to code for anything. Viroids don't code for anything. So I think it's a fine line, and it's a matter of nomenclature. Anyway, that's cool. I like that discussion. And so the upshot yesterday is that we're going to have a box in Chapter 1 where we discuss this fine line between viruses and plasmids, as we have discussed today.
Yep.
Cool.
This reminds me of an interview with Richard Feynman about names of plants and birds and that kind of stuff. And he says, what does it matter? It's a bird. Enjoy it.
Doesn't know its name.
The bird doesn't.
No, it's for us. It's always for us.
Right.
Did you see that crow? Did you see that?
Yeah.
And here in New Jersey, we have lots of goldfinches. It's a state bird, actually.
Is it?
Yeah, it is. And they're lovely. We have a goldfinch feeder. We get tons of them. And they're beautiful. Little yellow birds flittering about. So we name them for ourselves. And so that's why we name viruses.
So we can talk about
them.
Yep.
Yeah.
Mm-hmm.
I'm sure.
I have a squirrel feeder.
Linnaeus was
they're all squirrel feeders.
Well,
that's right.
Our bird feeders are basically squirrel feeders.
Yeah.
We have so many things. We put those metal things around the pole. The squirrels bloody jump from the roof of the house.
Yeah, the squirrels are great.
Have you seen chipmunks getting into them yet?
No, they get up
That's
there.
a hoot. The chipmunks, some of them can actually climb. I've got one of those that's supposed to bring a cage down when something as heavy as a squirrel pulls on it.
Yeah.
The chipmunks, some of them can actually climb on it, and it doesn't pull it down enough, and so they'll get into the seed.
What were you saying, Rich?
I had a wooden bird feeder once, and the squirrels actually ate the bird feeder.
Well, I mistakenly left a bag of bird seed outside.
Oop.
Then the squirrels chewed through it. Yes, Kathy.
I was wondering if there was a goldfinch in a cherry tree that was blooming. Would that be lovely or not lovely?
No, I wouldn't go see it.
No.
If a finch were in a cherry tree, would it be lovely? It's a great question. I like that. Is
it before or after I took my allergy medicine?
Right.
Does it make a noise in the woods if no one's there to hear it?
We have one more. Brianne, can you take that last one?
Sure. Angela writes, really cool TWiV. I'm a former virologist who works in DNA forensics. I always wondered if there were mitoviruses, or if mitochondria were evolutionarily related to viruses, because mitochondria replicate similarly to some viruses. An example is parvovirus. Now I know there are mitoviruses, and I have some catch-up reading to do. Curious if the viruses integrate into the host mitochondrial genome and alter the sequence. If they do, a mother and child might not have the same mitochondrial genome sequence, and or a single source profile may look like a mixture, and this could wreak havoc on the work that I do. Thanks for these great shows. Helps me keep up on my favorite topic of viruses. P.S. All the world's a phage.
Yeah, I don't believe that they integrate. Remember, they're RNA viruses, right?
Okay.
Single-stranded RNA viruses. So, and, integrating in the mitochondrial genome could be catastrophic.
Yeah,
it's small, and it doesn't code for a lot of proteins, so there's not a lot of places where you could go in. Although I guess, the bad ones, that mitochondria would be dead.
Yeah.
Right.
But that's you start messing up the mitochondrial genome, you can really kill your host fast.
Mm-hmm.
So
Yes, you can.
mitoviruses in plant chloroplasts, they have a much larger genome.
Well, we don't know.
Yeah,
That's a good question. We discussed that last time. Are there chloroplast viruses? I didn't look it up. Bradley, stop the car and respond.
Right.
Are there mitoviruses or chloroviruses of chloroplast?
Right.
Well, just based on the fact that chloroplasts are, somewhat related to cyanobacteria.
Yeah, there should be. Right?
Yeah.
Should be if there are cyanobacterial phages.
Yep. Yeah, that's what we think these mitoviruses came in with the bacteria when they entered cells, right? They're related to the viruses that were there.
That infect rickettsia and the other similar things.
Now, we have 2 papers today. And the first is a Journal of Virology paper. It's called Medusavirus, a novel large DNA virus discovered from hot spring water. And this is
open access. But
is it open access? Okay.
Yes.
And I think this is interesting. It's not by any means the biggest virus, but it has some interesting features that I thought you would like. And I really just wanted to pronounce the Japanese places. That's why. This comes from Kyoto University, the National Institute for Physiological Sciences in Okazaki, Japan. Isn't it cool that there is a town called Okazaki? Of course, yes. I think Dr. Okazaki probably was named after the town, not vice versa.
Right.
But I don't know. Tokyo Institute of Technology and the Tokyo University of Science. And I have to tell you, I was in Japan a couple of years ago in Tokyo, and Shinjuku is one of those divisions. What do they call them all? They're districts, the different, like Meguro and Akihabara, if you want to go buy electronics or so forth, and Shinjuku. And I used to call it Shinjuku, right? And then I run on the subway, and they have those automated female voices, and I just remember they would say Shinjuku, monotonous, but with the accent on Shinjuku, Akihabara. Okay. Pokemon. That's it for that paper.
It's a ward. It's a ward.
Thank you.
The only reason we covered that paper was
ward. It's a ward. Yeah, there are lots of cool wards with neat names and different things that you can see in each of them, and that's one of them. Shinjuku, Meguru. Okay.
Anyway, there's a paper here.
There's a paper here, and this was isolated in Japan from hot spring water, as the title says.
They have a lot of those.
And that's the connection between the 2 papers. They both involve hot water.
Yes.
This is, the genome is 300.
Hang on. You did the title and the institutions. I don't think
I
you
didn't
did
do the authors.
No.
Genki Yoshikawa is the first author. And do we have, 3 co-senior authors. So KMHO and the last one. Okay, so the co-senior authors are Kazuyoshi Murata, Hiroyuki Ogata, and Masaharu Takemura. And full disclosure, my son does take Japanese, so he corrects me all the time.
Your Japanese sounds kind of Italian, but that's
okay.
Come on.
A little bit.
Come on. Well, the next paper,
Masaharu Takemura.
The next paper is all Italian.
You got a problem with that?
We did these papers just for the authors. At 381,000 base pair genome, double-stranded DNA. So it's not anywhere near a big genome. In fact, that's close to the largest genome ficodinoviruses before the Mimi viruses were even discovered. But it's got some interesting properties, so I thought we would.
Just as comparison, the big giant viruses are almost 10 times the size of this.
Mm-hmm. Yeah. 2.4 million.
So this has a T equals 277 icosahedral capsid. So my question is, what's the largest T equals?
Oh, I have a.
Is that one of the giant viruses? Is it 1,000 something? Or?
I think, one of them is 1,000 something, if I remember. Yeah, they're huge. Oh, remember cafeteria?
Rowenbergensis.
Right.
Yeah, which we did not too long ago. Is that a big one? Let's look up the T number.
I did a search for largest viral triangulation number and came up with 219, but the paper is 15 years old.
Oh,
So, and I just Googled T number, cafeteria Rowenbergensis, 499
okay.
for cafeteria Rowenbergensis.
Nah, I'm pretty sure they're bigger ones.
Yeah.
Anyway, for those
Yeah,
of you
I
who
said-
wonder, this T number or triangulation number, it's the number of subunits per, or the number of facets per triangular face of the icosahedron. It's the simplest way to explain it. There are mathematical ways to derive it, but to make bigger, the simplest virus is at T equals 1, where you have a single protein repeated 60 times to make the smallest possible capsid. But to make bigger viruses, you have to stuff more subunits into each triangular face of the icosahedron, and that increases the T number. So this is really big.
Yeah.
But there are bigger ones, I'm quite sure. And I suspect there's an over 1,000, but I can't remember what it is. So this one is
So they took some muddy water that was leaking out of a hot spring.
took some muddy water.
So
and
I wanted to ask about this because I looked at how they did their enrichment and filtration, and it left me confused. So
Me too.
Okay, then
Yeah,
it's not just me.
they did a filtration twice that was supposed to do. I didn't understand which fraction was which. in
Okay,
that
right.
each time.
Yeah, so they do filtration through a 20 micron filter, and then the collected mud and dead leaves were resuspended.
So they
in
keep the part
sterile
that didn't
PBS.
go through the.
They
through
keep
the
the
filter.
stuff on the filter, right?
Right.
And stir it gently for a day, and sample again filtered through another 20 micron filter. Then
This
the filter,
time they take the filtrate.
it seems that way. They take the
Right.
filtered sample
Hmm.
through and put it through a 1.2 micron filter. And the filtrate was then mixed with medium and so forth and so on. So it just, so the 1.2 micron filter, and this thing ends up being 0.26 microns.
Yeah,
So it
went
should
through.
go through that 1.2, but it should have gone through the 20 micron filter
Right.
Yeah, it should have.
like crazy.
It
So
must have been stuck to the mud and dead leaves.
I
now that
guess.
we're talking about that, I'm just thinking maybe they were trying to separate free virus particles from virus particles that were in hosts. So the first time they're pulling out all the floating viruses and throwing that away with the filtrate, and they keep the mud and leaves where the amoebae would be.
Yeah.
Then
Yep.
they let that incubate for a day. Give virus. I'm just theorizing here, but this just occurred to me. Maybe what they're doing, and they don't spell this out in the methods, but maybe what that's doing, that stir it for a day, is to let more virus get out, then pull that out at the second filtrate, and then go from there.
I like that.
Could be.
Theory.
That's my story for now.
So they take it and put it on amoeba cultures. They actually are isolating virus, Acanthamoeba castellani, and that I can pronounce with an Italian accent.
And very well, I might.
And they see that this, whatever is there, is killing the cells.
And this Acanthamoeba castellani is just a standard laboratory strain.
Right.
of
Unless you
amoeba.
get it in your eye, and then you're in trouble.
Right, but it's something that's not necessarily in the environmental sample that they took.
Yeah. Although they say
Though we're going to get to,
amoeba are a potent tool to isolate large viruses, and but they say the actual natural hosts of these viruses have not yet been determined, which is true.
Yep.
Everyone seems to use amoeba, and who knows what they actually infect, although we're going to see a little evidence here.
Not to switch gears, but I looked through my virus structure lecture, and I have a slide about Medusavirus with its T number of 1179.
Mm-hmm.
All right, there you go.
Yeah.
Okay. Yeah, I wonder if there's a bigger one. Because Mimi is not the biggest, of course.
Right.
Of course, they may not be icosahedral. You only have T numbers with icosahedral viruses. Okay, back to this virus, which all right, so they infect these A. castellani cultures, and then a lot of the cells die from cytopathic effects, but they say some of them become encysted. They undergo morphological changes similar to encystment in a subpopulation, and others are lysed. And they say this encystment-like phenomenon prompted us to name this virus after Medusa, the monster in ancient Greek mythology who turns onlookers to stone.
So encystment is something the amoeba does under stress or dehydration to go dormant and survive for a long period, and this is happening in these cultures.
And now, of course, when this paper came out, a number of newspaper headlines said the virus that turns cells to stone, which of course is not correct. It does not. It's not stone. It's because of Medusa. Anyway, this is Medusavirus.
Medusa is the one, the woman with snakes for hair, right?
Snakes for hair.
Yes.
Yes,
if
one
you look
of
at
the
her directly,
Gorgons.
you turn to stone.
If you look at it, you turn to stone?
Yeah, that's the story.
You have to look in her eye or what?
I think Ulysses pointed a mirror at her and she saw herself and turned to stone.
That was smart.
I think so.
It's very clever.
So as well.
Very clever. If nowadays she could look at herself in her cell phone and turn to stone, right?
That's right.
So this the first thing is this is wonderful. They do cryo-EM to solve the structure.
Cryo-EM.
Single particle cryo-EM, and that's where they can figure out the T number. It's 260 nanometers in diameter, and it's a very interesting looking particle. It's covered with a number of spherical headed spikes, which extend from each capsomere.
Those are the snakes.
That's what I thought.
Very good. So I went over to the electron microscopy database where they deposited their coordinates, and I got a JPEG of the virus, which doesn't have any labeling on it, because the virus in the particle in the picture in the paper is labeled. So we'll put that as the episode picture. And that's going to be my pick of the week, by the way, or one of them. So this is a icosahedral particle, and it's interesting that underneath the protein shell there's a membrane, which happens in a lot of these nucleocytoplasmic large DNA viruses, of which this is one of many, including our friends, the pox viruses, the ficodna viruses, Mimi viruses, and by the way, and this had escaped me, the Pac-Man virus.
Yeah, I saw that.
Did you know there was a Pac-Man virus?
I do now.
We got to get them all, don't we?
I went to figure out why they were named Pac-Man, and they said because in a lot of their EM images, the capsids were burst open, and so they look sort of Pac-Man-like.
Oh, my gosh. And that's, of course, the Marseille group, so creative at naming. But here you see in Japan, they want to be equals. They want to name viruses cool things. So this virus replicates in the nucleus. It seems to assemble in the cytoplasm. They do some labeling studies where they can label the viral DNA. So you go into the nucleus and start replicating, but then the cytoplasm fills with capsids, and that's where they seem to be assembled. So that's where the name nucleocytoplasmics comes from, right, Rich? A lot of these viruses do both.
This is worth a little bit of comment because they have a really nice paragraph in the discussion that summarizes the different replication styles of this class of viruses. Some replicate entirely in the cytoplasm, some replicate entirely in the nucleus, and some spend part of their time in the nucleus and part of their time in the cytoplasm, and others, apparently, according to the reports, cozy up to the nucleus early after infection and cause export from the nucleus of replication enzymes. So it uses the nuclear enzymes in an extranuclear capacity and ultimately results in the destruction of the nucleus. So kind of anything goes. Some of those studies, I think, would be worthwhile to repeat with methods like they use in this paper, because this is really convincing. They use fluorescence in situ hybridization to look for where the DNA is. And there's absolutely no question whatsoever that this is happening. The most of the replication is happening in the nucleus. They also sort of consistent with this whole wrap. People talk about whether the viruses code RNA polymerases or whether they code DNA polymerases, and that sort of information can be used to help lend support to the notion of their replication style. If they have their own RNA polymerase and they package it in the virion and they encode their own DNA polymerase, that would indicate that it's something that does everything in the cytoplasm. If they encode an RNA polymerase, they may be able to use it sometime. If they don't have it in the virion, they can't initiate a replication in the cytoplasm, and on and on. There's all different types. But this guy has no RNA polymerase, right?
Correct.
And no DNA polymerase. Was that correct?
No, there's a B.
That's right. We're coming to that.
Yeah.
But the fluorescence in situ hybridization data is unequivocal. It replicates in the nucleus.
Yeah, it's quite nice.
And later exports stuff. Assembly occurs in the cytoplasm.
Yeah. So they sequence the genome, and there are 461 open reading frames. 3 tRNA-like sequences, so that's no big deal because there are viruses with all the tRNAs. What's cool here is that 86 of the predicted proteins had their closest homologs in A. castellani, suggesting massive gene exchanges between the ancestor of this virus and the amoeba. So that
in both directions.
Both directions.
Yes.
So clearly, this has been infecting A. castellani at some point. So they say this must be at least one of the hosts of this virus.
And it encodes its own histones.
Yeah, that's the cool part. That encodes a lot of histones.
Yeah.
More than any other virus. I think that's what caught my eye here originally. It's got all 4 core histones for the cognoscenti H2A, H2B, H3, and H4. And linker,
it has H1.
And the linker H1.
Or its own.
There are some viruses that have a subset of the 4 histones or fusions of histone-like proteins, but this is the first dude to show up with all 4 and the linking and the linker.
And they do proteomic analysis of the virus particles, and they find all the histones in the virus particle, suggesting that the DNA in the particle is wrapped around nucleosomes, which we know happens for other viruses like polyomaviruses. So, but here the histone
in that case, polyomavirus is borrowing host histones to wrap up its DNA.
Yeah. So this virus has its own. So I thought that was quite interesting. These histones in the virus particle. And the other thing that's interesting, the virus particle has 80 different proteins in it. Of course, there's the major capsid protein, but there's other stuff. There are other enzymes besides these histones. And 20 proteins that don't look like anything we have ever seen before. Orphans. And finally, if you do the phylogenetic analysis, it looks like this is a new lineage of DNA viruses. So they're proposing a new family, the Medusaviridae. So 57 candidates that have gone back and forth between Medusavirus and its host in both directions, as we noted.
Right.
Yeah,
Both
and there's
directions.
some interesting things that have been picked up by this virus in terms of cell biology, like a caspase and a nuclear import protein like RAN.
Yeah. So this is also the first giant virus from a heated environment.
Yes.
Here on TWiV we have heated discussions.
Yes.
But this is the first virus from a heat 43.4 degrees Celsius.
110 degrees Fahrenheit. That's kind of not scalding, right?
It's a warm bath.
It's a nice, I would, maybe even a hot bath.
Yeah.
It's the kind of thing you would sit in. There are lots of those in Japan, right?
Sure.
You can go sit in them because of the volcanic activity, yes?
Right.
And I think that is more or less it. These spikes on the surface, we don't know their function, of course, but they look very nice. And I love the colors. I'm just kidding. Of course they have no colors. Colors are assigned randomly. So here we have a new family proposed. We'll see if it's accepted. And it's from a hot source, and it has histones. It encodes all the histones, and it's got them in the virus particles.
Yeah.
And
this is good. This is the NCLDVs. We probably barely scratched the surface. This is going to keep going on and on.
You look and you will find, right?
Yeah.
Even Dickson could find new viruses nowadays.
Perhaps.
Dickson, if you could name a virus, what would you call it?
Oh, Vincent Sella.
Oh, come on. So, by the way, I drove to Albany last week to have a nice visit with Kara Pager, as in lager. That's how I remember how to pronounce her name. And on the way, I passed Coxsackie, New York.
Yes.
So I thought I should one day visit all the places in the U.S. that have viruses named after them.
Now, when you get to Neuro, do not go to Ohio.
Norwalk.
Don't go.
And you'll get to go to the Heartland.
That's right.
Yeah, the Heartland.
Yes. That's right.
Where am I going to go for the Heartland?
A lot of places.
A lot of places. So Four Corners and Norwalk and Coxsackie. Yeah. Maybe at each place I should do a TWiV, right? Lacrosse. There you go.
Yeah. St. Louis.
Lansing, Michigan. Poliovirus from Lansing, Michigan. Can we think of anything else? I wanted to run this by the TWiV crew because I knew this would be the right place to get them all.
You confined yourself to domestic. Of course, if you open it up to international, it's going to go on forever. Sure.
Yeah,
you're going to have to go to Rift Valley.
We don't have the money to go international. I could do a driving tour around
You could probably spring a trip to Sendai at least.
You go to Uganda and you can find them all there, right?
No, but what about U.S.? What else we have here? Well, you'd probably have to look at the list of viruses. Then it would jog your memory.
Eastern and Western.
And of course, while I'm at Four Corners, I have to go to Muerto Canyon. That was the first name, I think, right? This is what it could have been, Muerto Canyon virus.
Right.
Are there any viruses named after places in New Jersey? I don't think so. New York has...
You're not going to do all the flu strains that are named after places.
Right.
Right?
Which one? What do you got in mind?
H1N1.
No.
Got a California or...
No, that's too many. And that's not very interesting. Right? There's just too many of them. But the unique ones, Coxsackie is a cool one. I passed and I held up my phone and took a picture.
I always get this confused anyway. Q fever. That's like rickettsia, right?
From the town of Q.
Yeah.
Exactly. From the town of Q. Queens. I thought it was found in Queens. I think that's why they named it Q fever.
No. I had an interview with a guy who at Rocky Mountain Labs who works on Q fever. He talked about why it's named that, but I don't remember. Our next paper is a PLOS pathogens paper. I thought we would continue our arc of understanding the causes of Alzheimer's disease, specifically the implication that herpes simplex viruses are involved, which we have talked about on a couple of occasions previously. And this is an experimental paper done in mice, which addresses this, which I think is quite interesting. And, of course, it's from Italy. There's a number of institutions in Rome. One of them is not in the list here, but it's in the methods because something was done there. The Istituto Superiore di Sanità, which I've actually visited. And I always joke my host there. I said, "Where's the Istituto Inferiore di Sanità?" She said, "No, Vincent." Anyway, National Research Council. Università Cattolica del Sacro Cuore, the Sacred Heart, Fondazione Policlinico Universitario, A. Gemelli, IRCCS, Sapienza University of Rome. I always like that one, right? Knowledge, Sapienza, and the Telematic University in Rome. And the first author is Giovanna Di Chiara, and the last author is Anna Teresa Palamara. And there are lots of authors in between with lovely Italian names like Giorgia Napoletani, Virginia Proto, Domenica Donatella Lipuma, but no Vincent Racaniello.
There is only one.
No, they're actually Many.
Or are the others in jail?
If you search Vincent Racaniello, you will find the obituary of one who died out on Long Island a number of years ago.
Oh.
It's not me.
No.
He
One of the other Alan Doves had a blog about his brain cancer, and then he died.
So herpes simplex virus type 1. We have talked about in terms of Alzheimer's disease, this neurodegenerative disease. We have talked about evidence for finding transcripts of the virus in the brains of Alzheimer's patients. We have talked about how in culture systems, beta amyloid is induced in response to infection, suggesting that it might be a kind of innate response to infection. We've talked about epidemiological implications.
Also, the tau protein gets hyperphosphorylated, which is another
Alzheimer's
thing.
And there are epidemiological studies which suggest this. So here is a paper done in
So there's some circumstantial evidence that this might be related.
And of course, there's a clinical trial ongoing now to take people with early onset Alzheimer's, give them antivirals for 2 years and see if
like 8 pounds a day or something.
Yes, grams a day. Yes.
And though everybody is reasonably familiar with herpes viruses, we can remind the listeners that these have double-stranded DNA genomes and they're enveloped.
Yep.
They're neurotropic. They can establish a latent infection in sensory ganglia, and then they can periodically reactivate. And that's what becomes important here. When they reactivate, oftentimes that gives rise to the cold sores and blisters on the lips.
And HSV1 is your classic cold sore herpes virus.
Right.
So the idea here is that in people, occasionally, when you get reactivation, the virus gets into the brain and you have herpes encephalitis, which is a serious disease. And what we have gathered from the previous work that we've looked at is that there's some thought that some of these reactivations may lead to kind of either a mild or abortive infection of the brain. And if you get multiple such reactivations could predispose you to Alzheimer's. So
the impression that I'm getting is that there can be essentially a subclinical infection of the brain with HSV1 that can have both latent and very, very mild reactivation, but enough to, I think the conclusion from the last paper was that this induces a protective response in the brain that tries to tamp down the herpes infection, and it has the side effect of having some, call it neurotoxicity, ultimately in the form of perhaps Alzheimer's.
And so you get these repeated reactivations, and the idea is that after a certain number, then you begin to get these symptoms later in life.
And usually we associate those reactivations being under some kind of stress.
Right.
Yeah, like taking Brianne's exam at the end of the semester.
Exactly.
Just to be clear here.
Go ahead.
Just to be clear here, this is all still in the realm of we're not sure.
Yeah, for sure.
Except for the stress reactivation. That's something herpes viruses have been doing since before there were humans. They have herpes viruses that we talked about a while ago on an earlier TWiV that infect coral reefs, that when the reef is under stress, the virus reactivates. So this is something that they just do.
The association with Alzheimer's, this is an ongoing story. And
I always have a problem because I was taught at a young age that if you got herpes in the brain, you were really toast.
You're dead. Yeah.
But now we're thinking, well, there could be gray areas, so you have to revise that, right? Because you're
just a sprinkle.
Our colleague Daniel Griffin said, if you have herpes in the brain, you're dead. But that's obviously not the case. No. So we'll see. Anyway, this is a mouse model where they establish a latent infection and then they do periodic reactivations. And so they scarify the lip of the mouse, which is a
nice natural introduction process.
The lip of a mouse is small, so a delicate thing. And then they keep these mice for a long time, up to a year. And then to reactivate, they subject them to hyperthermia.
They dip them in a hot water bath.
15 minutes. They've sent them to Japan to the hot springs.
Yeah, the same temperature.
43 C, which is almost the same temperature.
Yeah.
It's pretty funny. And then this is so funny. After they take them out, They put them under a warm lamp so they don't get chilly
Yes.
for 30 minutes. Like
So they get a 15-minute hot bath, and then they dry under a heat lamp. It's a mouse spa. But this is a thermal stressor, which they're thinking will reactivate the virus, and as we'll see, it does.
Yes, yeah.
So do they get a masseuse also?
Ooh.
Very good, Dickson. Very good. Anyway, they do this every 6 to 8
This model,
weeks.
by the way, it goes back 20 years.
Oh, yes. This is not new.
Been around for a long time.
This is new, but they're using it very nicely.
Mm-hmm.
Yeah, they also do mention that they had to do some preliminary experiments, changing the virus dose and the way that they did inoculation so that they did not get the virus in the brain leading to encephalitis or seizures. So that goes back to that idea of it. Maybe it's just a little sprinkling.
Yes, absolutely. You have to keep the dose down. Otherwise, you don't want to kill the mice, right? And these mice will survive.
And we need to remind everybody that mice do lie.
Yes.
And crafting animal models for human herpes viruses has been a very, very, very difficult task. And so there's all kinds of ways you can bash this because we have a human virus in a mouse.
Right.
But, you know, a lot of what they're seeing here looks pretty good in terms of the reactivation and everything else.
Yeah, and this model does a really nice job, I think, of, to the extent possible, replicating what happens in humans, like the lip scarification.
Yes, exactly.
You know, we're not injecting this intraperitoneally, which is another way this is sometimes done. It's going in through the lip, like a herpes virus infection would in the human. And then the reactivation is this thermal stress, which, okay, you know, this is a kind of stress that can cause reactivations in humans, I think.
Mm-hmm.
But the idea is you're stress reactivating it. You're not giving them some compound that's going to do this. So it's a lot about it seems very realistic. And as we'll talk about when they test outcomes, they seem to be taking a pretty rigorous approach there too.
Yeah. So they do repeated reactivations every 6 to 8 weeks, up to 7 times, right? And initially they say, can we find virus in the trigeminal ganglion? And after reactivation, can we find it in the brain? And they look in a variety of ways. They look for genomes, they look for proteins, and I'm reading and I'm going, yeah, but do you look for virus? And yeah, actually they did.
They did.
So it was great. They look for infectious virus, both in the trigeminal ganglia and in the brain. Of course, those mice have to be sacrificed, so they don't make it into the long-term study.
Yeah, there are even plaque assays in here.
Yeah, and so they show you can you do this scarification of the lip, the virus becomes latent in the trigeminal ganglia. After this heat, this water bath treatment, it does go in the brain, and you can detect infectious virus in the brain.
Actually, I think it goes into the brain even without the stress.
Yes, it does. Yes.
My reading of this is that in upwards of 80% of the mice that they infect, they can detect herpes in the brain.
Right.
You know, several weeks after infection without any stress or anything like that. And it's those animals that they here it is, 83%. It's those animals that they wind up ultimately studying in the stress response. Okay, they specifically select out animals where they can detect herpes in the brain to look at what happens to that herpes when they undergo stress.
Right.
So that immediately brings up the question, when in a human, say primary infection, how many of those does virus go in the brain, right? We don't know.
No.
Yeah, this is something that would not be difficult to do, it seems to me, getting post-mortem samples of people, and
you'd have to get post-mortem samples of people right after they were first infected with HSV1, right?
It's tough. That would be very hard. Because
so
remember, we did
post-mortem samples from infants.
We did do a paper where they looked at older brains of people who died of Alzheimer's and normal controls, and they can find transcripts, but we want to look earlier because the question is, after your primary infection or even after reactivations, how often does virus go to the brain? We're under the impression that it's a rare event because we're looking at encephalitis, but maybe if it's a subclinical infection, it happens more often. So,
and the other question with the post-mortem analyses that have been done previously, people are looking at Alzheimer's versus healthy, and in the Alzheimer's brains, we've got more of the virus there. But then you get into the question of causality: is the virus there because they had Alzheimer's?
Yeah,
right, exactly. Is Alzheimer's really a situation where your brain is just more permissive to infection in general?
Rich, you were going to say something?
No, you've covered it.
Okay.
All right. So they extensively characterized this model. It's quite clear that you scarify the lip, the virus is latent in the trigeminal ganglia. Then on this water bath, it goes to the brain. So then they say, okay, let's look in the brains and see what happens. And they look at a variety of things that are related to Alzheimer's disease: the appearance of amyloid beta, the tau phosphorylation, neuroinflammation, and then a learning thing. A learning.
Yes, so they did. This
a
is
memory
this
test.
Is
I forgot what it's called.
Challenge.
Right. So I was reading the novel object recognition paradigm (NOR). So I'm reading along in this paper, and I'm thinking, okay, this is all lining up nicely. You've got the plaques and you've got the amyloid in the brain, you've got the tau hyperphosphorylated, you've got inflammation, but what's happening to the mice? Did you take them to the opera? Right. And then you get to memory loss induced by recurrent HSV1 infections. It's like, okay, yeah, they took them to the opera. Specifically, what they do is they put the mouse in a cage that's got two objects, and I put a link in here that actually has a nice video and everything showing how this test is done. So you have two identical. Go in
the show notes. It's really amusing, especially the video in that link.
Yes. So you've got two identical objects, two like little cubes or something, and you introduce the mouse to it, and they'll investigate both cubes for a period of time, and then you repeat this experiment where you swap in a different looking cube, and they should investigate that for longer because it's different. Mm-hmm. Unless they don't remember this from before, in which case they'll investigate both of them equally. And so you can quantify the degree to which they remember having seen something. And you find with this that the ones that have gotten the virus and that it's been reactivated, with the reactivations, they are less able to remember.
And they compare the third heat shock with the seventh heat shock, and it gets worse. With increasing, and there have been fourth and fifth and sixth in between. So the more you get, the more you forget. And they do the yes, Kathy, go ahead.
Well, sorry, this is really kind of back off the subject, but I was thinking about the paper that we did a couple weeks ago from Mariah Spara and Matt Weitzman looking at HSV2 in the infected neonates, where they had to work so hard. And they ended up with 10 samples and isolates and so forth. But overall, it looked like, or they said that HSV causes invasive disease in half of infected neonates. So it's just one number, and getting at Rich's question of how could you do this? Figure out the frequency.
So the amyloid beta they look at in these mice following the reactivations, and it accumulates clearly after increased reactivations. Tau phosphorylation and cleavage occurs, all in the right portions of the brain. I'm skipping over a lot of the data. It's really nice data, but we don't need to belabor
lost pathogens. It's open access. So
cleavage of tau by caspase, which is a marker of neurodegeneration, and then signs of neuroinflammation, astrocytosis, activation of astrocytes, the production of pro-inflammatory cytokines. All of these are up in mice that have undergone multiple reactivations. Yeah. So all of that is consistent with Alzheimer's, right? And this is caused again by reactivation of herpes simplex.
And they have right near the end of the paper, they've got a nice graphical summary. Figure 10 nice. that summarizes the whole set of findings and claims, including a baffled Alzheimer's mouse trying to figure out which object they've seen before.
So there could have been another set of experiments in this in which they heat shocked them and gave them an antiviral at the same time.
Yeah, that would be good.
Why didn't they do that?
Or they could just give them the antiviral continuously and then do these heat shock things, and that's going to prevent this. I'm sure that would, most likely, right?
Maybe that's.
Although someone wrote us 2 weeks ago and said the drug does not penetrate the brain very well, valacyclovir.
Right.
So that could limit.
Might have to inject it intrathecally.
Oh, yeah.
Then
Sure.
what?
No,
How are
that's-
those mouse brains going to be?
Well,
Yeah, and then it's not realistic for what you would be doing in terms of a real treatment.
So now the big question with this is, if HSV1 infection is somehow causative or exacerbating in Alzheimer's disease, why doesn't everybody get Alzheimer's disease?
Yeah.
right.
Because herpes virus infection, just about everybody's been infected with at least one herpes virus, probably multiple ones, right?
Yeah, for sure.
So
Good
why is only a subset of the population developing Alzheimer's disease?
Mm-hmm.
And I guess one possibility that this paper raises is with this stress reactivation is maybe it's a function of how much reactivation you get, and or other stressors that have occurred, because there's also another thread of Alzheimer's research looking at physiological and other factors that are causing inflammatory responses and this sort of thing in the body, which could all feed into this pathogenic process. So it may be that the more of those you have adding up, the more herpes virus you have going to the brain and causing this, or something along those
I wouldn't
lines.
be surprised if there were polymorphisms in, say, innate
too.
immune genes that normally would keep the level of virus down, so it maybe doesn't go in the brain. And in some people who have change in TLR3 get more herpes replication, more invasion, something like that. So people can take Alzheimer's genomes and do the GWAS, the genome-wide association studies.
If there is a small enough number of cofactors that you can actually tease it out that way.
Right.
I think,
and if you can get patients that are sort of homogenous enough.
Yeah.
Right.
I also think there are obviously other causes of Alzheimer's. So there's a bacterial, a mouth bacterium,
Porphyromonas
gingivalis, implicated in a recent science paper. And so there are probably other environmental things as well.
Yeah, I think this is one of these diseases that's overdetermined. It's like obesity or high blood pressure or all these other things. Well, what's causing it? Well, all these things are causing it. And I think it's going to be the case that this is a pathogenic endpoint. You can get there by many routes.
Now, the other thing I thought of is, okay, what if any virus got into the brain over and over again? Let's say you found a sublethal dose of poliovirus in a mouse, and you just multiply injected 6, 7, 8, 9, 10 times. You don't kill the mice, but enough to get virus in the brain. Would they develop amyloid beta plaque formation, tau phosphorylation? Would they forget in their learnings? Is any virus that goes in the brain going to have the same reactivity? Why does it have? Herpes simplex, it's latent and keeps getting reactivated, and it's in the trigeminal ganglia, so that's a good one for that. But maybe in principle, any virus getting into the brain could do this.
Right.
Do people with West Nile encephalitis have an increased incidence of Alzheimer's? I don't know. So it may not just be herpes, right? It may be, but that one is the one that does recurrent brain infections, whereas the other viruses like polio gets in, and that's pretty much it for you because it's an acute infection that then you make immunity to, et cetera, et cetera. Well,
but now there's a mouse model where one can study the antiviral processes and everything else that's going on with this. So this sets up a really nice set of experiments where you can go in and look at what is going on in these mice immunologically in those brain cells when you reactivate the virus.
Or look in mice with different TLR3 mutations.
Yes. Or put in different,
Yeah. So the last sentence of the paper, our findings strongly support the view that recurrent HSV1 infection is a risk factor for AD.
That's well said. A risk factor.
Sure.
Strongly support the view. They don't say it proves it.
Right.
Strongly support. And I would say, with all the stuff we've seen now, including this, it's looking very interesting, isn't it? I'll be really curious to see the results of that clinical trial.
Yeah.
Wouldn't it be something if an antiviral could be useful for Alzheimer's disease after all the research that has gone into, cellular causes and so forth? Because it's a big deal. A lot of people biggest cause of dementia in humans, right? AD.
Maybe there's an approved generic drug that can treat this. That would be big news. Yeah.
One thing that I was just going to comment on methodologically, they call their plaque assay standard plaque assay, and they abbreviate it SPA.
SPA.
S-P-A.
Yes, I noticed that.
They also use a technique that they call in-cell Western. And I kept looking at that. Finally, I said, isn't this just immunofluorescence? So then I finally went to the paper that they reference for that. And evidently there's something called the in-cell Western assay from Lycor, the company that makes the scanner that allows you to measure this immunofluorescence. And yes, it's basically an immunofluorescence assay, but they call it a Western assay. To me, would imply that there's blotting involved, and that's not here.
Yeah.
Or at least gel separation.
Yeah.
So. Anyway, nicely done work. It's all carefully done, beautiful results. Everything has been considered pretty. I like the paper very much. So there you go.
Yeah, me too.
You like it because it's AD.
Yes, right.
AD. Let's read a couple more emails. Rich, are you there?
I'm here.
Can you take Anthony's?
Sure. Anthony writes, Dr. Condit mentioned that to be appreciated, rats need to grow fur on their tails. It'll take more than that. People view subterranean animals with disgust and arboreal animals with awe. Nocturnal animals are feared, while diurnal animals are accepted. And he gives a link to a wonderful PDF that talks about I'm blanking on the
Stephen J. Gould.
Yes, Stephen J. Gould. It quotes an article of his on the historical evolution of Mickey Mouse and how Mickey Mouse went from more of a rattier looking creature who was up to all sorts of nasty tricks to a mousier looking and creature who was nice because of, he became such a national icon that he had to be a good guy. And they had to look really cute. And that morphogenesis, according to this article, has to do, among other things, with becoming more baby-like in appearance. Yes. Okay? A stubbier snout, a bigger head to body ratio, shorter, fatter legs, etc. And the bottom line is that people warm up to babies. So things that look more baby-like we like it better. Says the biggest factor, though, are the proportions of the skull. The elongated rat snout is a reducto ad absurdum of that of the adult human, and so poses a threat. The pushed-in nose of the squirrel resembles a human infant's skull shape, and so elicits a response of attraction and caring. For what it's worth, Anthony. Anthony's always got some choice observations for us, and so
and those are good points. I would though point out that mice have fur on their tails and are considered cuter than rats.
Yeah.
They're otherwise very similar. They have the elongated snout. They're very, very rat-like in their appearance, but fur on the tails would definitely help rats. It may not make them as cute as squirrels because of the skull shape, but fur would be a start.
Oh, so that's why giant microbes makes fuzzy viruses, right?
Right, to look cuter.
Yes.
Did you know Stephen J. Gould Dickson?
I did. I actually met him.
He went to Columbia, apparently.
He did. He was a geologist and an evolutionist by training. A big Yankee fan.
Yes.
Yankees as in the Northerners?
No, as in
the baseball team.
Oh, that.
Baseball team.
Okay. Alan, can you take the next one?
Sure. Anonymous writes, and I assume this is a person who wishes to remain anonymous, not the hacker group known as Anonymous. Dear TWiV, TWIM, and Immune, the measles issue ain't going away because you have chiropractors making rants, and links to a few chiropractors ranting about vaccines. There's a convention called California Jam. It's like an ASM meetup, but filled with chiropractors and anti-vaxxers, according to one of these posts. One of the people in the video is named Heather Wolfson, a chiropractor. She's one of the players who keep spreading the anti-vax rants in a consistent manner. The problem here is that she's identified to the public as a healthcare provider who has a medical degree, even though chiropractors and MDs are separate entities. And then provides a link to the I guess this is Rockland declaring measles emergency and barring unvaccinated kids from public places. Yeah. So good points.
All right. Sally writes, hi, you mentioned archiving issues. Well, the TWiV 68 plaque assay movies are dead links for science, and I can't find the fluorescently tagged late early stage visualization of a plaque assay that you described in recent episode. Can you help, or all three unavailable now? Thanks. All right, I fixed the link to the plaque assay movies. You know, journals just change their links from time to time, and I can't go back and check them.
This is a problem. This is a worldwide problem with the internet, and I read a really disturbing article about this where in a lot of cases legal opinions from the courts, all the way up to the Supreme Court, and this is internationally, other courts, anybody who uses English common law, it's all about precedent. So, the legal opinion of the court is the law. And in modern cases, they are often referencing websites by putting in URLs. And so you get these legal opinions that 5 years later, you can't click the link anymore. It's gone.
Right.
Yep. So our legal code is corroding by link rot.
Link rot.
Ew.
Well, thank you for writing, Shally. We appreciate it. Anyone else find some link that's not working, let me know, because I do fix them. Although the other movie, the recent one, which was sent in, it was a 35 megabyte GIF, and it was too big to upload. So I'm sorry, a number of people asked for that, but I just.
But you fixed the TWiV 68.
Oh yeah,
links, right?
Oh yeah. Yeah, science movies are still there, and I fixed those, so that's great. Brianne, can you take the next one?
Sure. Angela writes about the Wascally Viruses episode. Thanks for the great show. Couldn't stop laughing about your comments. It's too GC rich. In the lab I worked in as a graduate student, we used to say this all the time when our PCRs of HSV1 genes failed. Those failures helped me learn how to troubleshoot PCR reactions. At my current job, when I mention GC-rich sequences in primers used for sequencing mitochondrial genome regions, people look at me like I'm crazy. Hearing that comment brought back some great memories. P.S. I think the authors, when researching why the plaques were smaller, forgot an area to look at: the packaging of the viral genome into capsids. Perhaps capsid formation and/or packaging of the viral genome was not 100% efficient, hence the smaller plaques.
Could be.
Sure.
Could be.
Kathy, can you take the next one, please?
Sure. Anthony writes, apparently he wrote a letter to Dorit Reese asking why isn't intentionally exposing a child to disease prosecuted as abuse. If you're asking whether there are laws that can be used, for example, against parents who take kids to measles or chickenpox parties, there are such laws. You would need political will to use them. There is prosecutorial discretion in the U.S. I suspect it would take serious harm to get them used against white middle-class parents. Unfortunately, our criminal justice system is not always even-handed. And the answer was from Dorit Rubenstein Reese, professor of law at UC Hastings College of the Law. So this is what Anthony sent us, having corresponded with her.
Interesting.
So
I assume
it could be prosecuted as abuse, but given the demographics of the anti-vaccine movement, it probably won't be.
And I don't really know whether Dorit is a he or she. Sorry, I made that assumption.
Rich, can you take the next one?
Sure. Raphael writes, Dear Alan, Dickson, Kathy, and Vincent, and everybody else. I was listening. It was actually, this is a specific show. I was listening to your show on the Science 360 podcast. You mentioned that both Europe and the U.S. have reduced antibiotics use in livestock production, while the developing countries are still using plenty. While being true, of course, it startled me a little, as at least here in Europe, there is a significant difference between the amount used per country. Of course, there is also a difference in the population of livestock or humans, but adjusted for that, many of the larger countries still use far too much antibiotics. My point being, I found your comment to make it easy for us developed citizens to feel good about our livestock production, while we really should be more aware of what impacts our choices have. Germany, being in the upper middle segment of livestock antibiotics users and with a very strong conventional farmers union, has a problem, especially in the industrial pork production. It apparently is that bad that pig farmers in some areas of the country going to the hospital for anything will be put into the infectious disease department of a hospital to protect the patients and staff from MRSA and similar. So I believe that we should be highly aware of how our food decisions impact this issue, and that it is still highly relevant in the U.S. and Europe as well as the developing countries. And then he's got a link for the EU report source. Thanks for the interesting podcast, Raphael.
Yeah.
Well,
and I don't know if I implied that the U.S. and Europe were all peachy, that was not my intent at all. There is still plenty of room for improvement. The point that I think I intended at least was, at least we're moving in the right direction. And there has been progress, more so in Europe than in the U.S., but in both places there's been progress toward reducing antibiotic use in agriculture, and we are doing less of it. I will add that the pork industry is a problem here too, for antibiotic resistance and other things. But in terms of the magnitude, the developing countries are using vastly more antibiotics, and it's much, much harder to get a grasp on in many of those settings because you're dealing with all sorts of logistical hurdles and legal hurdles and questions of you can't just go in and tell people what to do when they're going to be starving if they don't grow enough food. So the issues are complex. Yes.
All right, let me just summarize the last two here. Anonymous writes again, Dear TWiV, we have another case of a
political... Could be different Anonymous.
I think it was the same from what I could tell. Of a political activist going after a real virologist over vaccines. Dr. Peter Hotez from one of the TWiV episodes has been targeted by Del Bigtree, a political activist, in a rant. So Hotez was interviewed by Joe Rogan, who is a YouTube personality with a big following, and talked about reasonable things, vaccines, and his book and so forth. And then apparently Del Bigtree, I don't know who Del Bigtree is, but he says
he's CEO of an anti-vax group called ICANN.
He made a video saying that Hotez shows why anti-vaxxers are right, which is crazy. So Anonymous sends a bunch of links to these. After Rockland County, New York banned unvaccinated children from public spaces last week, as it battles an outbreak that originated in an Orthodox Jewish community, anti-vax activists likened that public health measure to the Nazi persecution of Jews. In Austin, Del Bigtree, CEO of the anti-vax group ICANN, wore a yellow star during a rally last Thursday to identify with parents who declined to vaccinate their children. A spokesman for Bigtree said he took the action to let the Jewish community of Rockland County know he stands with them. After photos of Bigtree were posted on Twitter, the official account of the Auschwitz Memorial and Museum in Poland tweeted in response, instrumentalizing the fate of Jews who were persecuted by the hateful anti-Semitic ideology and murdered in camps like Auschwitz with poisonous gas in order to argue against vaccination that saves human lives is a symptom of intellectual and moral degeneration.
Well put.
Jonathan Greenblatt, CEO of the Anti-Defamation League, said groups advancing a political or social agenda should be able to assert their ideas without trivializing the memory of 6 million Jews slaughtered in the Holocaust. Texas has been one of the most organized and politically active anti-vaccine groups. Texans for Vaccine Choice, which organized a rally last week, received support from Tea Party Republicans and some of the state's most influential conservative organizations. Hotez, who has been an outspoken critic of the anti-vaccine lobby, called its latest actions highly offensive. and asked the leadership of the state Republican Party to publicly denounce its actions. He added, I don't know why they think they gain by mocking the Holocaust.
Right.
The rise in measles cases was both predicted and predictable, Hotez said.
What he stands to gain is to compare vaccination to Nazi experimentation.
Right.
Okay. Nazi atrocities. Mm-hmm. He's basically saying that vaccination is like a Nazi atrocity.
It's like the rule that any argument that goes on the internet for long enough, eventually somebody will be compared to Hitler. Yeah.
Right.
Well, the difference, of course, is that vaccination saves lives, not an experiment.
No, exactly. It's there's no comparing them. This shouldn't even be a conversation. And it's this guy is just, this is the way he operates. And, he got a lot of attention for this, right?
Sure.
Rich, you were at this rally, right?
I was at a different rally.
Okay.
I was, so this is worth talking about though. I went to a rally on Tuesday, April 2nd. That was a collaboration between Immunize Texas and Ginny Sue's group. I think her group has a different name. I'm blanking on it, but it was a rally in support of vaccination that coincided with a thing called First Tuesday, which when the Texas legislature is in session, which is only, I think it's 3 months every other year. So it's pretty.
We don't need your stinking laws.
It's going on. It's going on now. They have First Tuesday where doctors from all over the state show up at the Capitol and lobby for various health issues with the legislature. And I learned at this rally. It was a nice rally. It was well done. And I actually met a TWiV fan there who was a mom with 8 kids. And is an avid fan. As a matter of fact, one of her kids is autistic, and another one had a very bad reaction to vaccines. But she was there to promote vaccination despite all of that. Right? She was a very nice, very interesting person. At any rate, I learned at this thing that the anti-vaxxers, while Texas legislature is in session, show up at the Capitol twice a week and go around and lobby legislators. So they're making a lot of noise. And the point was made that they are yelling so loudly and making so much noise that they wind up convincing some that they represent a larger fraction of the population than they actually do, which is their goal. Jenny shows up at least once a week, I think on Wednesdays, and has a table in the rotunda, I think. So the Immunize Texas and other groups are trying to make a comeback to these activities, but it's a real struggle.
Rich, you should go.
I should go see what Jenny's up to.
Yeah, because we need voices there.
That's right. I should go. As a matter of fact, I was not completely informed, or I did not inform myself. I could have been more informed about this rally. I thought I'll just go to the rally. But in fact, it was organized so that after the rally itself, there were breakout groups that went and lobbied legislators. Okay. And on another occasion, I will do just that. And I also ought to go visit Jenny and see what she's doing.
Our last email is from Ryan, who says, Dear TWiV, we have a measles scare reported in Sacramento, largest in California so far. Sends a link to an article where someone with measles went to an emergency department and may have, exposed quite a few people. And this is what's going to happen. That's why you can't be in public and not be vaccinated.
Mm-hmm.
But yeah, if you do have measles, it's appropriate to go to the emergency department, but we shouldn't have people with measles.
Right.
That's right.
Well, that begs a question. If you have measles and you're suffering, the ER should be able to
handle it.
But if you're sitting there in the triage area before they know you have measles, then you can infect other people, right?
Yes.
So it's kind of it's a tough situation.
Very true.
Well, let's do some picks of the week.
Mm-hmm.
Brianne,
excuse me just a second. Jenny's organization is Immunize Texas.
Right.
There is another organization that was also represented there that I will try and find the name of that has a similar mission.
All right. Thank you.
Sounds good.
Brianne, what do you have for us?
Well, I think many people probably saw the picture of a black hole that came out this week. And the woman who was the team leader, who has been getting a lot of publicity, did a TED Talk a couple of years ago about how she was doing the algorithm that was being used in order to take this picture of a black hole. And so Katie Bauman, that researcher formerly from MIT, has a whole TED Talk about the algorithm that was used in order to make that picture that I think we've all seen this week.
It's a great TED Talk. It really explains it. It's only about 12 minutes long, and it's so worth it to watch it. And I'm so glad that you found this, because I saw this stuff on Twitter about New York Times. Why didn't you even mention the woman that was involved in this whole thing? Oh. And this is great.
I've seen all of my social media has been all about her and different aspects of her fame and women in science and things like that this week. And I thought actually hearing her talk about the work and how it was done was a nice way to credit her.
Yeah.
Yes.
Yeah, earlier today, she was on the front page of the website, but since has disappeared from it.
She was featured prominently on the NPR show about that. Yeah.
One of the reasons she's become so famous so fast, besides the fact that she did this amazing thing, is that there's this picture of her with this face of the joy of pure discovery. As it's wonderful. It's just like, yes, that is what science is. Those are the moments you live for. And so that's part of it. But it's amazing work.
It's like finding the Higgs boson particle. I think that whole group is going to get the Nobel Prize the next time for physics.
So this photo of her with the amazed look has been circulating on Instagram, along with a 1960s photo of a woman standing next to a stack of paper, which was the code needed to get to the moon.
Right.
So another lady did that programming.
Sure.
Right. Very cool.
Yes.
Very cool stuff. And it's wonderfully dated. It's just great. Alan, what do you have?
Knowing that probably somebody would pick the black hole image as their pick, this has been in my list of bookmarks for a few weeks, and I thought I gotta do this as a TWiV pick. This is an image from NASA data. So a citizen scientist, an amateur astronomer, Kevin Gill, took data from the Juno spacecraft and stitched it together into a complete image of Jupiter, and it is freaking gorgeous. And if you click on the picture and you zoom in, you get the full size image, and it's just this beautiful marble, this swirling Van Gogh looking Starry Night type of image. And the as you look at it, try and imagine the scale of what you're looking at. The big red spot and the smaller storms. These storms, you could put Earth inside them.
Mm-hmm.
And this is just floating there in the blackness of space. And I thought it was a really cool image.
What blows my mind about these is that they are so turbulent and yet stay there.
Yes.
I don't get it. Somebody does, I'm sure, but I don't.
Yeah, there's a whole fluid dynamics thing going on with the winds on it and how they establish these eddies. And I don't know either, but that is a very interesting phenomenon that you've got these century-old storms that stay put.
Yeah, it's pretty cool.
Yeah, well, remember how big they are.
Yes.
So they're not staying still, but they look that way, that's for sure. You know, when I look out the window, for instance, and I'm talking with Vincent, and we're doing a TWiV or a TWiP, and I see airplanes coming in to land in Newark, they look like they're going to fall out of the sky. They're not moving. They're so clumsy, and they look like they're going to lose aerodynamics and everything else, but they're traveling at 180 miles an hour. So if you get the closer you get, the faster they look. So this is pretty far away. So I'm going to tell you that this is chugging.
Rich, what do you have for us?
First of all, let me back up with a final correction here. The groups that did the rally are Immunize Texas and the Texas Immunization Partnership. Immunize Texas being Jenny Sue's organization. Okay, so I have another nested pick. This I had picked out when it was a little more immediately relevant around the time that we were in Iowa, but we didn't do picks then. So a nested pick in that there are 3 picks here. The first, I like the primary. This is all about the commercial crewed spaceflight program that is going on now. And it's called the NASA Commercial Crew Program. And I linked to the NASA site for that, which you can make your way through several links there and get roughly what the program is about and get updates and et cetera. But there, I found what I like better, although it takes you 30 minutes to get through it, which is a YouTube video from a guy named Tim Dodd, who runs a podcast called The Everyday Astronaut. And I later link his YouTube page as Everyday Astronaut. And this is a guy who is just a space nut and is very good at describing this whole thing. And I wasn't really aware that NASA and the government have awarded are funding or helping fund 2 commercial manned spaceflight programs, one by Boeing and the other by SpaceX. And they're both developing their own capsules that have different characteristics that are to be mounted on rockets with different characteristics. And the one that's most advanced at this point is the Dragon capsule from SpaceX, which a few weeks ago made its first unmanned trip to the space station and went up and successfully docked. And they sort of went through a whole mock thing of getting in and out of the spacecraft. It has one advantage it has a fairly large cargo space. So I think it actually carried up some cargo on its test run. And then successfully undocked and returned to Earth. So this is a nice description of that whole program.
I don't know how this guy gets to go into these things. He's inside them. How does he do that?
Yeah, I don't know. But what his story was that somebody gave him an old used spacesuit at one point, and
Really?
yeah. And it turned him on. He went nuts. I guess he was already nuts to start with.
What's most amazing to me is how these rockets land on Earth after they come back. They just these skinny things with the little fins. Oh, yeah. They go
gyros.
Straight down.
There's a lot of gyros,
and they don't fall over.
Yeah, balancing.
Gyroscopes.
Pick up a ladder in one hand and balance it there. That's what's going on.
Right.
The SpaceX crewed capsule, the Dragon, was initially the initial concept is that capsule itself with people in it would do the same thing. It would have a powered descent. And there were issues with that. So at this point, that design is not what's going forward. Yes, the rockets that take them up will do SpaceX's thing of landing and being reusable, but the capsule itself is gone to splashdown. And the Boeing has limited, if at all, reuse. The Boeing capsule is going to be a hard landing, but like Soyuz.
Boeing probably isn't calling it a hard landing.
No.
But it's going to be on land rather than the sea.
Yeah, that too.
Dear.
Dickson, what do you have for us?
Well, everybody's got their head in the clouds, it looks like, because I've got some shots from the International Space Station showing what they call sky-high stunners. Rich, I put this in there just for you, by the way, because I know you're almost as crazy as the everyday astronaut, and I'm just right behind you on that. So, I go to NASA's website probably every day and take a look at the Juno mission for sure, and just wonder how all that stuff works is exactly right. But this one is, take a good look at some of these pictures, like the first one showing, that's in Bangladesh, that's the Brahmaputra River exiting, and if the ocean goes up 2.5 to 3 feet, most of that will be gone. The next one is of the Delta in Louisiana, and that it's almost gone now. and another 2 feet, it'll be gone as well. So a lot of these have some significance because they're sort of historical views of things that will no longer be as the Earth continues to change.
There's a nice one of penguin poo in Antarctica.
Yeah, that's exactly right. Exactly. You eat enough krill, and they'll be able to follow you no matter where you go.
These are great.
Yeah.
Kathy, what do you have?
I picked 2 photo contests here at U of M. One is the Arts at Michigan Photo Winners, and I especially like the first one.
I agree.
Once you click on it, then there's a sideways arrow from it to all the rest of them. But it's a black and white of the Grand Canyon that just has the most interesting perspective, light and shadows. And then a lot of the photos in this exhibit or whatever are black and white, and I thought that was interesting. So check out those images. And then the other one just came up today because somebody sent me the second place winner of the U of M Microscopy Core Image Contest. And it's a picture using rabies to identify neurons. And so it doesn't have a link to a paper or anything like that, but the figure legend describes what you're looking at. And they only sent me this number 2 in, and I said, wow, if that's number 2, I wonder what's number 1. So I went back and checked out number 1. So they're just several nice microscopy images.
Number 1 is
very
beautiful.
Cool.
Beautiful.
Yeah.
I would like to know where the tough cells are there.
Yeah.
They're up in Boston, Dickson.
These are so wonderful and so descriptive and yet so informative at the same time. It's no wonder we were attracted to this as people. Really, I mean, we're all in this for different reasons. But when you see something like that, the beauty of it just absolutely, you want to see more.
You're in it for money, right, Dickson?
Absolutely. That's why I
see
went.
how that worked out.
Yeah, you do, actually. I'm not telling you everything, Alan.
Oh, right.
I have 2 picks. One, I'm continuing my picks of Beth Mole articles over at Ars Technica following this New York measles outbreak. She has an article on New York City declaring an emergency and mandating vaccination. You can't be in public unless you're vaccinated. So this is in parts of Williamsburg, which is in Brooklyn, where a community has had a big outbreak since last October among unvaccinated children. So if you're in a certain zip code, you have to get the vaccine or you can't be in public.
How
and
would
props
they know?
for using a picture of a smiling kid getting a vaccine.
No, Vincent.
Thank you.
I know, Dickson. I was thinking the same thing. How would you know? No. You have to receive it or prove immunity. I don't know. You can't prove immunity. You're going to have some kind of certificate saying that
show them your antibodies. Come on, show them your
any unvaccinated people living in infected zip codes who may have been exposed will be required to receive the vaccine or prove immunity. So I think this is very hard to execute, but maybe it will get people to vaccinate.
So, Leonard,
it'll improve rates. You won't be able to get everybody, but this should improve rates.
That's right. So there was an NPR radio show that I was listening to about 2 days ago where the city health commissioner was on, and someone was saying to them, they were phoning in, and a lot of very Orthodox Jewish leaders from Williamsburg phoned in to say, listen, we're going to tell you something now. Anybody who says that they're not vaccinating their children for religious reasons, and if they're Jewish, there is no religious tenet for that. There's nothing written anywhere that says because your religion says this, you can't do that. The vaccination wasn't even known about when the Old Testament was written. So these people are making it up as they go along. They just don't trust science. Remember, they don't trust science.
Well, they cannot argue against the science any longer.
They cannot.
So they move to other things.
Exactly.
Religious exemption is one.
Right. Exactly.
The idea of I should have a right over what to put in my body is another argument, right? And then they say we should get immunity naturally with natural infections, right? That's another one. It's just all nonsense.
It is.
Unfortunately, I see in this article.
You do get immunity with natural infections.
You do.
That's why we made vaccines in the first place.
Exact.
And the immunity is excellent with a vaccine without the pathogenic consequences.
Right.
Also in Rockland County, that was the last pick I made of Beth Moll's article. They had banned unvaccinated children from public spaces, but a state judge apparently halted that ban, so they're trying to figure something out. That's unfortunate because you have to reach to the legal system, as we saw from that letter.
Yeah,
from the lady.
I have a thought here. What if all the vaccines that are available today had a little bit of vaccinia in it also? So that when you were vaccinated, you could show that you were vaccinated because you've got a scar. And then, I've got it. See,
I don't
here's my arm.
That's quite the right approach, but
I prefer.
There's got to be some marker.
Here's my idea: put GFP in, and then you could just see if
everybody
bingo.
Shine a little UV on, and if you're glowing, you don't even need UV. In the sun, you probably have enough. Just
exactly right.
Because the fish that are GFP transgenics, they're green without fluorescent light.
Well, these are the same people that want to eat GMOs for the same, so let's not do that.
Yes, Kathy.
Well, so I have two things. If the government has the ability to quarantine,
they do.
How is that different from the ability to, not allow people into public spaces? Number one, or to require people to get vaccinations if they want to go out in the public. Okay.
Yeah.
And then the second thing is that Washtenaw County, which is where Ann Arbor is, just got an epi alert today about measles. An international visitor to the county has been confirmed with measles. They flew into Detroit, developed symptoms and rash during the visit, flew home, and were diagnosed in Germany. And so this case is not part of the current Michigan outbreak and had no recent measles vaccination. So,
right.
Yeah, so it's just a list of all the places where you can go and get vaccines.
And one of these big issues also relates to the fact that these kids were not allowed to go to school that hadn't been vaccinated. So they've been home for a long time now, and they've missed a lot of school. So it's crazy all the way around.
It's a solution. They could get vaccinated and go right back to school.
They could go right back.
And you know what? You'll be fine. Nothing's going to happen except you'll be protected. What is the issue, folks? Right. But of course, everyone listening is understanding.
The issue is a belief system that we've talked about.
That's right.
My other pick is the EM data resource. So we have talked about the protein data bank where you can deposit coordinates of protein and virus structures. Here's another one for electron microscopy images that can be low resolution images or quite frequently, like the one today, the cryo-EM structure of Medusavirus is deposited here. So these are the coordinates which you need to have a viewer to look at it. That's not so easy. But with each entry, there is a little JPEG which shows you the structure, and it's very nice. And you can go through and see all the viruses and the big molecules that they're solving structures of. And they're, on the front page here is the Racaniello SH1 vertex, and they have big enzymes. So here's the full particle of another virus. Here is the contractile injection system of a bacterium, an enzyme. It's full of cool stuff.
Yeah,
ribosomes,
bacteriophage, icosahedral capsid. So
that's a little known counterpart of the protein data bank.
I did a search for virus. I did a search for a virus, and there are 2,000 hits.
Yeah, lots of viruses in there.
Yeah.
We have a listener pick from Martin. Maybe a mention of Sidney Brenner's passing might be a podcast pick sometime soon. So Sidney Brenner passed away. He sends a link to the Telegraph article, which is not accessible. I have a link to a Nature article, which, I don't know, maybe, Alan, can you see if
let me just check that.
I was able to do the Sidney Brenner one just by just putting in my name.
It's totally readable with no login whatsoever.
Okay. So it's a nice... You got the Telegraph also? Okay.
Yeah, and the New York Times obituary was nice. I had that earlier this week. So...
Yeah, so Sidney was a major force in molecular biology, and these articles will tell you all about what he
was.
Did.
A worm guy.
He was, although he started out as a molecular biologist. He worked with Francis Crick for many years.
He did.
And
credited with co-discovery of messenger RNA.
Mm-hmm.
Wow.
That's a big one.
Yeah.
And then Dickson mentioned worms. He pioneered the C. elegans system, where, for those who don't otherwise know, what's it got? 900 some odd cells in the body of this worm,
Right.
including a nervous system and muscles and a reproductive system. And Sydney realized that you could potentially map the fate of every cell in the worm from a fertilized egg to the mature animal, and that's what his lab did, and that was the origin of his Nobel Prize. There's a genetic system there as well, so it's a marvelous developmental biology system.
Yeah.
He was described as one of biology's mischievous children, the witty trickster who delights in stirring things up.
Right. He was fun to listen to.
He was amusing. He gave, the only time I heard him talk was at Rockefeller when David Baltimore was being installed as president. And he said, when I first got the invitation, I thought I would come over with some tools and pipes and put them all together, and that would be what my installation would be. But no, this is something different.
This is the Telegraph article saying he had a passion for words and wordplay. In a riff on Avogadro's number, he invented avocado's number, defining it as the number of atoms in a guacamole. Guacamole.
Guacamole. Very nice.
Yes.
All right. Too bad we never had him on one of our podcasts.
92 years old.
He would have been fun to have on TWiP.
Hey, I have Elio Schechter on TWiM, who's 91, or maybe maybe he's 92. I don't know.
Yeah.
All right. So that is TWiV 543. If you like what we do, please consider supporting us financially. We do have expenses, and we'd love help with them. And we'd also like to do more things. Go to different cities and do podcasts and see all of you, but can't do that at the moment. So you can find different ways to help us out. Microbe.tv/contribute. No matter what you decide on that, we always love hearing from you. Questions and comments, TWiV at microbe.tv. Dickson Despommier can be found at fordemopolis.com and thelivingriver.org. Thank you, Dickson.
You're welcome, Vincent.
I hope your voice gets better.
So does my voice.
So does your voice.
Yes, it certainly does.
Brianne Barker's over at Drew University on Twitter. She is BioProf Barker. Thank you, Brianne.
Thank you. It was great to be here, and I hope you feel better soon, Dickson.
Thank you. Brianne, didn't we just hear some good news about you?
We did, yes.
Right. We forgot to mention this.
No, we didn't. I just mentioned it.
Yeah, very good.
Yes. The committee that decides on tenure decided that I got tenure on Wednesday. It will be official with the Board of Trustees in about a month, but my application has been forwarded and voted positively.
Outstanding.
Excellent. Outstanding.
See, that's
Congratulations.
the TWiV push. This is called the TWiV push.
The TWiV bump, yes.
Kathy Spindler's at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor at University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
You can find Alan Dove at turbidplaque.com or on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I am Vincent Racaniello. You can find me at virology.ws. I want to thank ASV, the American Society for Virology, and ASM for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.